So this is a short video demonstrating how membrane filtration sterility testing is performed in the Bioquell Qube isolator. Firstly, the operator prepares the environment by placing a settle plate at the back of the cube. So you can see here they are writing the details such as the date and time that the settle plate was placed. And once these details have been written, they will take the lid off of the settle plate and then they will place it at the back of the cube on the working tray or the floor before starting any processing. Next, the operator will open the Merck-Millipore test kit, which as you can see contains two canisters, one to detect aerobic growth and one to detect anaerobic growth. They will place these two canisters inside the pump apparatus and then place both of the tubes through the peristaltic pump, which allows liquid to be drawn through the tubes. So the next step is to connect these tubes to a bottle of rinse fluid by puncturing the needle through the top and then also puncturing an air filter in to allow air to replace the fluid that's taken from the bottle. Then the pump will be activated to draw this rinse fluid through the pump and into the two canisters so that the filters inside the canisters are moistened. Now the next step is probably the most critical in the process. It's where the sterile medicinal product is actually filtered into the two canisters. So as you can see, the operator is breaking off the top of a glass ampoule, and then they are putting the filling needle into the glass vials one by one, and they will eventually filter the liquid from 20 separate drug samples through the pump's tubing and into both of the canisters, which will collect this drug product, and the filter will become absorbed with this drug product. Now, this is the highest risk of introducing contamination into the process because the drug vial or ampoule is technically open to the environment, and this is why we are doing it in the Bioquell Qube in a closed isolator. So once the drug samples have been pumped into the two canisters, the next step is to actually pull the drug sample through the filter. So what the operator is doing now is placing caps on top of the two canisters and running the pump. And what this will do is actually pull the liquid from inside the canister through the filter and then into the waste tube that you can see off the right-hand side of the two canisters. And with the drug product removed from the two canisters, the next step is to again introduce rinse fluid into these two canisters. And this additional rinse step is needed to remove any drug residues from the filters, which may have an antimicrobial effect and kill any contamination that may have been caught by these filters. At this point, the operator will now seal the two canisters to ensure that when additional liquid is added, it will stay inside the canisters. That's what these yellow bungs are. They're sealing off the canisters. So once both canisters are sealed, the operator will take one bottle of growth media for aerobic organisms and then one bottle of growth media for the anaerobic organisms. And then they will begin to pump the first bottle of media into one of the tubes, one of the canisters, in order to fill it with growth media for aerobic growth. Then when the operator runs the pump, you will see the liquid starts to flow from the bottle through the pump and into the left-hand canister, and eventually over time the canister will become full of growth media, as can now be seen. Once the first canister is full, the operator will remove the needle from the empty bottle of growth media, transfer it to the second bottle, and then they will close the valves to ensure that the second bottle of growth media is transferred to the empty right-hand canister. And again, the pump will be run to fill this right-hand canister up with the other type of growth media until it's completely full. So with both canisters filled with the two types of growth media for anaerobic and aerobic growth, the operator will close off the two valves, the red and white plastic pieces here, and then they will cut the tubes below these valves to completely separate the two canisters from the pump apparatus. Now these two canisters can be removed from the pump and collected for incubation. With the process now effectively complete, the operator just has to pack down and remove the materials. So first they will remove the discarded tubes from the pump, and they will collect all of the waste into the packaging, which was brought into the Qube to begin with. They will then transfer all of the waste from the process out of the Qube through the QMTD, the material transfer device on the right, by opening the internal door, sliding the waste materials in, first with the Merck Millipore test kit, then with the empty drug vial carrier, and finally the critical samples, which are going to be incubated for 14 days. So once the operator closes the transfer door, the Qube is available to start the next test.